Hi, this is Professor Tiffany Martin with Medical Microbiology, and we are in Chapter 7, Microbial Genetics. And the last thing in this chapter that I want to talk about is something called horizontal gene transfer. Now, we've talked about DNA, we've talked about replication of DNA, and when we're replicating DNA, the point is to pass it on to our progeny, right? Just like grandma's cookbook, and you want to copy it so that you can pass it down to the next generation. So, replication involves copying it and passing it down. That is called vertical gene transfer from mother to daughter, from one cell to all of its progeny. That's vertical gene transfer. Prokaryotic bacteria are capable of doing a horizontal gene transfer. They actually have a couple of different ways that they can pass DNA to their neighbor. Not their progeny, but their neighbor. And this you don't see in eukaryotic cells, but you do see it in prokaryotic cells. So let's talk about this phenomenon, horizontal gene transfer. The donor cell contributes part of its genome. And again, when we're talking about horizontal gene transfer, unlike DNA replication where we make a complete copy, this is just sharing a portion of their nucleic acids, not the entire genome. So it is different than vertical gene transfer when you would pass everything down. So there's three types: transformation, transduction, and bacterial conjugation. And this conjugation might sound familiar because we've kind of already covered this in chapter 3. So let's skip ahead to conjugation and do that one first. Conjugation, do you remember that pilus? When we talked about a pili, where it can form this structure between neighboring cells. We talked about that. It's a specialized form of fimbriae, and it involves genetic transfer. And we said what gets transferred is not the chromosome, but this plasmid. So if you go back and review that, you had an F positive cell. It's F positive because it contains a fertility factor, and then that fertility factor will replicate itself and then cross over using that pilus, and now this cell will become F positive. So we talked about that, F positive and F negative. The pilus will only form with an F positive cell, will only form a pilus with an F negative cell. We talked about it getting replicated and then crossing it over. Didn't really mention that before, but there actually is some movement in this when that pilus is formed. It actually brings it closer. For that, this process, this crossing over of the plasmid into the new cell is called conjugation. That is one way we get horizontal gene transfer in prokaryotic cells. All right, so let's go back into the other two. There is another process called transformation. Transformation is when you get horizontal gene transfer, but instead of a plasmid, the cell picks up DNA from the environment. So maybe you had a bacterial cell that died. It got chopped up, either a drug killed it or, phagocyte, whatever reason. You have pieces of nucleic acid that are in the environment. You have a neighboring bacteria cell that is able to take up that piece. Now, most cells don't do this. Most cells, the cell membrane, cell walls, keep stuff out. We don't want random pieces of DNA and getting into our cell in general. It's a lot more chance that things will get messed up than it's not. But there are a few bacteria cells that are, or species, that are naturally capable of picking up pieces of DNA from the environment. So it's just an adaptation that they have, a survival strategy. By picking up pieces of this in the environment, then they can pick and choose which ones they want to keep. Cells that can do this and take up DNA from their environment are referred to as competent cells. So again, not all cells can do this, but there are certain species that are naturally competent, which means they have the ability to pick up DNA from the environment. Now, unlike a plasmid, this little piece right here gets incorporated into the chromosome, and then it stays with it, and then it can be passed on to their progeny by vertical gene transfer. Okay, so this happens naturally for a few species. However, as scientists figured out how this worked, it triggered a train of thought going, what if I could make a cell pick up a piece of nucleic acid? And it wouldn't necessarily be a random piece from the environment, but maybe this is a particular gene that I'm studying. and it's found in people and it causes some sort of disease, or I think it does, but it's hard to study things in people. What if I take this gene and put it into maybe a yeast cell or a bacteria cell, and then I can study it better? Or maybe this is a gene that produces insulin. And we know that patients who have diabetes, if they take insulin, they can live full and healthy lives. If they take this insulin, well, it's very hard to make enough insulin to give patients who are diabetics. What if I can make like a bacteria cell produce insulin? If I can take this gene for insulin and get it to integrate in here, and then this particular cell will pump out a whole bunch of insulin protein. That's a good thing. So there are reasons where you can do that. And this is called recombinant DNA technology, where we can use this process um, and get a cell to take it up. Now, once that cell has taken up the DNA, we refer to that cell as being transformed. So that's where the name comes from, transformation. Once it has taken it up, if a cell can take it up, it's called competent. Once it has taken it up, it is now referred to as a transformed cell. Okay, so that's another type of horizontal gene transfer. Happens in nature, but we have taken that theory and made it work for us. Like things like producing insulin. All right, so one interesting thing about transformation, how did they figure out this happened? So going back in time to like chapter 1, 1800s, early 1900s, there was a scientist living, his last name was Griffith, and he did some experiments and he proved that transformation could occur. He was looking at these microorganisms. There was a particular one that they had identified called Streptococcus pneumoniae. So Streptococcus pneumoniae um, actually could cause pneumonia in patients, but it didn't always. And when they grew it on a plate, on an agar plate, the same bacteria could actually grow two different ways in the colonies. It could produce colonies that were some strains of Streptococcus pneumoniae would produce very smooth colonies, and some strains of Streptococcus pneumoniae would produce these rough, kind of undulating colonies. And so they looked different on that. And when he looked at them under the microscope, he found the ones that produced these smooth colonies had a capsule, and the ones that produced the rough colonies did not have a capsule. So he named the strain that had a capsule, he named that strain S for smooth, and the ones that didn't make a capsule were strain R for rough colonies. So we remember capsules can be virulence factors, right? One of the things that happens with a capsule is that it makes it hard for your immune system to grab a hold of it, right? Purpose of a capsule. So the strain R cells that did not have a capsule, when they were injected into mice, the mouse's immune system took care of it and the mouse lived and didn't get pneumonia. If you took strain S with the capsule and injected them into the mice would get pneumonia and the mice would die. So strain S was lethal, the ones with the capsule. Strain R was not lethal. It lived. Now, he did do one other thing in preparation for this experiment. If he killed strain S, so heat treated dead cells strain S, and he injected them into the mouse, well, then obviously these are dead. They didn't cause pneumonia and the mouse lived. Okay, so this kind of set up this experiment. These were observations he made. And so his experiment was, what happens if you inject living strain R and dead strain S into the same mouse? What would happen then? Now, hypothesis, strain S, mouse lives, strain R, dead strain S, mouse lives, dead living strain R, the mouse lives. Your hypothesis could be that the mouse is going to live. So let's do the experiment and see what happens. So living strain R, heat treated dead strain S, neither one of those by themselves are lethal, injected to the mouse, and now the mouse dies. From pneumonia. He cultured the bacteria that he found in the mice. And remember, these are rough colonies and these are dead, so you would get no colonies. What he saw was smooth colonies. So he saw living strain S. This this Streptococcus pneumoniae now is making capsules. So how did they do that? So a couple of different thoughts. you could, maybe they took the capsules from the dead ones, and just kind of appropriated them. But the numbers were too big. We had too many bacteria making capsules. So they had to take the instructions from the dead strain S, the instructions on how to make a capsule, and incorporate them. And now they have the ability to make a capsule. So that's what he's found out. So he found it in vivo in a mouse. He also found it in vitro in a test tube. He saw the same thing, the heat treated dead S and the living strain R, and he saw some cells were able to take up the instructions, and now he had capsule producing Streptococcus pneumoniae. So that's how they knew that this was happening, even without being able to see it happen. All right, so we've done transformation. We've done conjugation. The very last one is transduction. Where do I have that? Transformation, conjugation. There it is. Transduction. What you need to know about transduction is it involves a virus. And we're going to look at this in a little more detail in the viral chapter. But for right now, this is a case of horizontal gene transfer where parts of bacterial DNA get transferred from one bacteria to another via a bacteriophage, which is a name for a virus that infects bacteria. Bacteriophage, phage is just another name for virus. Bacteriophage is a virus that infects bacteria. And you can see you've got a bacteria cell here. This bacteria cell is getting invaded by a virus. The virus is going to kill off the nucleic acid and chop it all up, kill off the cell. It's going to make a whole bunch of more viruses, and then it's going to lyse and release these virions, these viruses, out into the environment. How transduction occurs is you get a little bit of bacteria DNA that gets packaged, and it brings it to the next cell. And instead of integrating the next cell with viral DNA, it infects the next cell with bacterial DNA, and then that gets incorporated into the chromosome, and this is where you can get horizontal gene transfer. We'll look at a little more detail in that. That will bring us to the end of genetics. I know it's a big chapter, but stay with it. And this is big. Chapter 5 and Chapter 7, metabolism and genetics, are the two biggest challenges. So hang in there, do it. You can do it, and I'll see you later. Thanks, guys.